Good morning, good afternoon, and good evening, everyone. Thank you for joining us, and we are very happy to have you for this webinar organized by Proteogenics. We are excited to dive into navigating the complexity of a stable cell line development together. So first, we will hear from Laura Radu, account manager at Proteogenics, who will guide us through the initial stage of a stable cell line development, and then Andrea Guth, senior director for stable cell line portfolio at Advanced Instruments, will introduce the VEPS technology and its role in the cell line development process. So now I will hand it over to Laura Radu for the first part of the today's webinar. Laura, it's your turn.
Thank you, Ines, for this nice presentation. Welcome, everyone, and thank you for joining our webinar today. It is a pleasure for me today to present you this webinar on navigating the complexities of the stable cell line development. Today, we'll focus on the vital role of stable cell line development in antibody production for biopharmaceutical development. CLD is essential for generating antibodies and recombinant proteins, ensuring consistent, scalable, and cost-effective production, which is critical for your project success. Selecting the right cell line and developing a robust CLD process lays the groundwork for future manufacturing and regulatory approvals. We will discuss the key steps in CLD, including gene insertion, screening, and selecting high-producing clones, all of which are crucial for success. The goal is to establish a cell line that demonstrates high expression levels of the target protein while maintaining genetic stability and scalability. We'll cover optimization techniques and technologies involved, such as selection markers, automation in clone selection, and robust screening processes. Challenges are inherent in any CLD project, including low productivity, stability concerns, long timelines, and regulatory complexities. I'll share strategies to address common issues, troubleshoot productivity, maintain stability, and avoid development bottlenecks. While advances in bioprocessing technologies such as automatic cell screening and high throughput analysis, overcoming these challenges is increasingly feasible. Finally, we'll explore how to advance your CLD project after initial development, focusing on transitioning from early stage to large-scale GMP manufacturing and preparing your cell line for clinical and commercial production. I'll highlight the importance of collaborating with a CRO that has expertise in stable cell line development to accelerate timelines and minimize risks. As we begin today's session, I'd like to introduce you to Proteogenics, which is a leading CRO in biologics discovery and development. Our company has a rich history starting from the discovery of a novel protein, ICVP90, in 1997. Fast forward to 2003, we established our commercial operation in Strasbourg, France, and today we've grown to 160 employees and have over 21 years of expertise in advancing biologic research. Our services cover a full spectrum of biologic discovery and development, including gene synthesis, peptide synthesis, polyclonal antibody development, monoclonal antibody development, protein production, and end product distribution. So Proteogenics is your trusted partner delivering scalable, high-quality solutions from gene to end product to support your research and clinical goals. Now that you know about Proteogenics, let's explore our comprehensive pipeline of antibody services, which offers an end-to-end solution from discovery to bioproduction. This seamless process ensures our customers don't have to coordinate multiple vendors to bring their therapeutic antibodies to life. Antigen production, design and production is the first step crucial for generating high-quality antibodies. So for that, we provide peptide synthesis, gene synthesis, and protein expression. Next, antibody generation includes polyclonal production and hybridoma development for monoclonals. We also use single B-cell technology for highly specific antibodies and phage display for difficult targets. After generation, antibody analysis follows, ensuring quality through epitope mapping, affinity and stability evaluation, and purity assessment. For sequencing needs, we offer hybridoma sequencing and de novo antibody sequencing by mass spectrometry. For the antibody engineering stage, we enhance therapeutic potential through affinity maturation, antibody humanization, and AI-based developability improvement, and the creation of bispecific antibodies and antibody-drug conjugates. Finally, during bioproduction, we ensure efficient, high-yield production with stable cell line development and transient mammalian expression systems, including high-throughput capabilities and large-scale productions. So by partnering with Proteogenics, you ensure speed, quality, and consistency throughout the entire antibody development process, saving time and reducing the risk of errors or delays. So the path to bringing biologics to market is complex and involves several critical stages, with stable cell line development playing a key role throughout. First, the process begins with in vitro and in vivo testing, where fundamental research and target validation helps identifying the right molecular targets for drug development. Once a candidate is selected, we enter the preclinical development where CLD becomes foundational. This stage focuses on developing stable cell lines that produce the biologic compound consistently and meet regulatory standards for safety and efficacy. Next, during the clinical development phases from 1 to 3, the biologic undergoes human testing. So CLD is essential here for preparing the investigational new drug submission to regulatory authorities, allowing critical trials to commence. After successful trials, we move to the regulatory review phase, where agencies like the FDA assess the data to determine if the biologic can proceed to market. At this stage, the stability and scalability provided by the stable cell line remains crucial. Finally, once approved, the biologic enters the post-marketing phase, which includes ongoing surveillance to ensure its continued safety and efficacy in real-world use. So in summary, CLD is fundamental to many of these stages, providing the foundation for reliable, scalable, and regulatory compliant biologics production. So while navigating the complexity of the stable cell line development, several key aspects need to be taken into consideration. The first one is the choice of cell line to be used. Selecting the right cell line is a critical step in stable cell line development, as it directly influences the production scalability and efficiency of the biologic. After cell line different cell lines have specific advantages and drawbacks, depending on the nature of the therapeutic protein or antibody to be produced. So key factors such as post-translational modifications, like glycosylation, for example, play a vital role in ensuring the biological functionality of proteins, particularly in biopharmaceutical applications. CHO cells is one of the most widely used cell line in biologics production, especially for complex proteins and antibodies. It has a well-established history for producing high-quality, scalable protein yields. CHO cells are often the preferred choice for large-scale production of monoclonal antibodies due to their ability and adaptability to perform human-like PTMs. However, they may require more extensive optimization to achieve the desired levels and the stability. HEK cells, while less common than CHO cells, are often used for specific types of biologics, including viral vectors. Its ease of genetic manipulation and human origin can be advantageous, but it may not be as scalable as CHO for large manufacturing. For animal health applications, where cost effectiveness may outweigh the need for complex modifications, other expression systems can be used, such as insect cells. So these insect cells offer flexibility for producing certain proteins, particularly those that require complex post-translational modifications. However, they may not always be the first choice due to differences in glycosylation patterns compared to mammalian cells. E. coli is also sometimes favored over CHO cells as they offer more affordable and faster production options with different optimization priorities. Ideal for producing simple proteins, E. coli offers high yield and fast production. However, it lacks the post-translational modifications that may be needed for more complex therapeutics, such as glycosylation. So choosing the correct cell line is a balancing act between factors like scalability, production efficiency, and the biochemical properties required for the biologic. This decision is crucial as it will impact downstream development and manufacturing stages. Genetic instability is a common challenge also in stable cell line development and can significantly impact the consistency and quality of biologic production. This issue can arise due to several factors like gene silencing, unstable gene integration, disruption of regulatory elements, and accumulation of mutations. These issues manifest in several ways, for example, reduced protein quality, decreased cell viability, and inconsistent expression levels. To address these issues, careful vector design and cell screening protocols are essential to ensure long-term stability and high productivity. Optimizing these elements early in the development process can mitigate many of these risks and ensure a more consistent and reliable production line. Achieving both high yield and long-term stability is often a trade-off in stable cell line development. Clones that produce high levels of recombinant antibody may be more prone to instability due to metabolic stress or other cellular processes. Conversely, highly stable cell lines may produce lower yields, but their long-term performance is more reliable. Successfully managing the balance between stability and yield is crucial for the production of biologics, such as monoclonal antibodies, enzymes, or other therapeutic proteins. This balance can be achieved by carefully considering key factors like the developability study of your sequence of interest prior to starting the CLD development in order to identify and exclude liability sequences that could be problematic during manufacturing, like glycosylation, oxidation, hydrophobic patches, or fragmentation risks. Also, careful selection of clones that demonstrate both high productivity and genetic stability is essential. And this is typically achieved through high throughput screening technologies and long-term performance monitoring. Cell culture media composition plays also a critical role in cell health, productivity, and stability. So optimizing the nutrient content and other components of the media can significantly improve the performance of the stable cell line, supporting both higher yields and great stability. Also, consistent and precise control over the production parameters helps maintain cell line stability. Any fluctuations in these parameters can lead to stress, instability, or reduced productivity. As we dive deeper into the development of stable cell lines, one of the most complex and crucial steps is also the clone selection process that includes proof of monoclonality, high productivity and stability from one batch to another, and of course, cost efficiency. In general, we can highlight two broad approaches to clone selection. The traditional methods, like limited dilution, where the clone selection relies on manual processes, and FACS. These methods involve significant hands-on effort, are time-consuming, and often introduce variability. While these techniques have been foundational in stable cell line development, they come with challenges in terms of efficiency and scalability. Recently, more advanced technologies have revolutionized this space. High-throughput automation, imaging-based technologies, and single-cell cloning platforms like VIPS have greatly improved the precision and speed of clone selection. With these advancements, we can now achieve higher monoclonality assurance, more consistent productivity, and faster development timelines. The result is not just a reduction in the time to market, but also improved overall quality and reproducibility of the cell lines. As we continue discussing the complexity of the stable cell line development, another crucial aspect to consider is the substantial investment in both time and resources. So the process of developing a stable cell line is inherently complex and time-consuming, often involving multiple phases of optimization and troubleshooting. For companies or researchers aiming to move their therapeutic or diagnostic product to market quickly, understanding and managing these resources is crucial. There are three key areas that heavily impact the efficiency and success of the process. The first one is specialized equipment, from bioreactors to high-throughput screening platform and analytical tools. The equipment needed for stable cell line development must be highly specialized. Another one is optimization and involves fine-tuning various parameters to ensure that the cell line exhibits the highest possible productivity and quality. And let's not forget expertise that is involved in equally important. Navigating through complex challenges such as managing gene instability or lower protein production requires a team of highly skilled scientists. So the development of a successful stable cell line often hinges on this deep knowledge and the ability to anticipate and resolve potential hurdles. So by investing in the right combination of specialized equipment, optimization strategies, and expertise, the process can be streamlined in order to maximize time lost to trial and error and ultimately deliver high quality cell lines that meet regulatory and market standards. In stable cell line development, regulatory compliance is not just a box to be checked. It is a critical pillar to ensure both quality and safety throughout the entire process. So it is important to provide rigorous documentation at every stage of the development process to ensure product approval and the foundation for transitioning into GMP. This can be achieved by using advanced technologies that ensure accurate clonality verification and providing detailed reports that are essential for regulatory reviews. So navigating this complex landscape of compliance can be challenging, but it's absolutely essential for bringing safe, effective therapies to the market. So before considering launching into a complex project like stable cell line development in-house, there are 6 key factors to evaluate. Expertise. So do you have the in-house expertise in stable cell line development? Is this something you already did before? The process requires a broad range of expertise, including genetic engineering, cell culture in suspension, and adherent cells purification using different methods like affinity, ion exchange, gel filtration, characterization, and multifactorial analysis. The second aspect to consider is technology. Do you have the necessary state-of-the-art platforms in place to develop and characterize your cell line? Because this requires extensive equipment like the lab setup, machines, VIPs, BioCore, HPLC, DSF, mass spectrometry to gather data, and trained personnel also to perform the analysis and information, to perform and analyze the information and make informed decisions. Did you already consider optimization of your sequence of interest before starting the CLD process? Because anticipating developability problems right from the early stages of R&D is a significant asset for the proper development of your project. Can you afford the risk of failure? A cell line that expresses little to none of the biomolecule intended for production can jeopardize the viability of the development project and possibly resulting in its discontinuation. It is well known also that speed to market is a critical competitive advantage in biologics. So how much time do you have to finish the project? Is it several weeks or several months, or is it 1 year? Can you afford the delays when you are under short timelines for patent deposition, for example? So as your project progresses from research to full-scale production, scalability becomes a crucial factor also. So do you have the capability in-house to scale up the production of your antibody of interest? So when you consider these 6 factors, it becomes clear that the development of a stable cell line is a strategic step into the process of developing a new antibody, as it plays a crucial role in the final cost and quality of your biomolecule. So once you've made your assessment, if only one of these criteria is missing, then you need to start thinking about outsourcing your complex CLD projects. This will allow you not only to benefit from specialized knowledge and resources, but you also reduce timelines and the risk of failure, helping you bring your biologic product to market faster and more efficiently. This is why partnering with a CRO that has all the previous criteria is the best solution, as it provides you with expertise, because CROs bring significant expertise to the table. They have vast experience in developing stable cell lines for various therapeutic applications. Another major benefit of working with a CRO is the ability to provide guarantees. They can commit to delivering specific performance metrics or timelines, helping you manage project risk and ensuring predictable outcomes. When it comes to timelines, partnering with a CRO can significantly accelerate your project because they have established optimized workflows and dedicated teams that help shorten the time needed for stable cell line development, allowing you to meet important milestones faster. Developing stable cell lines requires specialized equipment also that is often costly to acquire and maintain in-house. So CROs have already state-of-the-art facilities, ensuring that you gain access to cutting-edge technologies without the capital investment. Finally, while it may seem counterintuitive, CRO partnerships can help you control costs. By outsourcing to a CRO, you avoid the substantial expenses of building and maintaining the required expertise, infrastructure, and equipment in-house. This allows you to focus your internal resources where they are needed the most. So in conclusion, by choosing a CRO for your CLD projects, you can gain access to a wealth of expertise, guaranteed outcomes, shorter timelines, advanced equipment, and controlled costs. Together, these advantages can give you a competitive edge in bringing your product to market efficiently and successfully. Now, let's dive into Proteogenics' streamlined CLD workflow, which provides a clear pathway from the initial needs assessment all the way to the scalable biologics production. This workflow is designed to ensure that at each critical stage, decisions are made with the highest level of scrutiny, guided by scientific rigor and data-driven assessment. A classical project begins with a stable pool generation, where the primary focus is creating a pool of cells capable of reliably producing the biologic. This is where we evaluate and select the best performing pools, ensuring scalability and consistency in the production. Step 2 involves single cell isolation, a critical phase where individual clones are selected for their ability to produce high yields of biologic with the desired characteristics. Monoclonality is ensured using VIPS technology with precise and efficient clonal screening. Using VIPS reduces selection time by 50% compared to other methods like limited dilution, for example, and also ensures confident selection of top monoclones. So my colleague Andrea from Advanced Instruments will present you the technology and advantages of using it for stable cell line development projects. Next, we focus on cell line characterization to ensure stability and high-quality biologic production. Here, we do a deep analysis to assess key parameters such as productivity, stability, and overall performance. Finally, step 5 brings us to the final production ready stage, where the clones are prepared for cell banking and scalable production. This is where the process is fine-tuned and preparations are made for the transition to full-scale GMP production. So these are the usual steps of a classical CLD project. At Proteogenics, we found that some of the most crucial steps occur even before stable cell line development begins, starting with a deep understanding of the customer needs. We invest significant time in learning the project's background, objectives, and specific requirements. This enables us to design and refine a customized roadmap that aligns perfectly with the desired goal. Additionally, we conduct an in silico manufacturability analysis of our sequence of interest to identify potential liability motifs such as glycosylation, oxidation, deamidation, and isomerization. We also detect hydrophobic patches and fragmentation motifs that could impact the solubility, aggregation, and stability of your candidate. A comprehensive report is provided detailing the identified risks along with the optimized sequences to mitigate these issues, ensuring an ideal sequence for use of the stable cell line development. The proposed variants are transiently produced and characterized, and the results are discussed with the customer. At this stage, a decision will be made regarding the choice of the lead candidate for the next steps. So throughout this workflow, we ensure that at each step, there is a clear go-no-go decision point. This allows us and the customer to move forward only when the data supports the decision, ensuring efficiency, scalability, and reliability of biologics production. Customers have the option to stop or pause the project at key points during the process in order to reassess the results obtained and next steps to be taken. So following, I would like to present you the results we obtained for a recent project of stable cell line development. So a leading biotech company approached us to do a stable cell line capable of consistently expressing a therapeutic antibody. The project goal was to move from transient transfection to the generation of a robust, reliable research cell bank, followed by characterization to ensure long-term antibody production. So the project began directly with transient transfection to rapidly produce the target antibody and confirm its functionality, as the antibody sequence had been optimized previously during affinity maturation process. To our surprise, the expression yield obtained was ridiculously low. It was around 6 milligrams per liter. However, the QCs confirmed the high purity and integrity of the antibody. So with the transient transfection results validated, we proceeded to create a stable cell line using our proprietary CHOK1 with cells with the objective of generating higher expressing stable cell line that could consistently produce the antibody. So we used a classical approach starting with gene integration. The gene coding for the antibody was stably integrating into CHO cells using our optimized selection system. We screened 5 times 96 clones, so around 500 clones, to identify the highest antibody producing pool. So our high throughput screening system in 96 well plates helped streamline the selection process. Then we selected high producing pools that were expanded, and antibodies were characterized for integrity, stability, and aggregation. And 3 high-producing pools were identified with yields exceeding 2 grams per liter in fed batch cultures. So at this point, the customers were thrilled with the improvement in the production yield and decided to continue the project with monoclonal selection by VIPS. So starting from the best expressing pool, monoclones were isolated using VIPS technology in 96 well plates, and expression evaluation was performed with ELISA with anti-FC antibodies. So Andrea will give you more details regarding the VIPS technology and how it is successfully used in stable cell line development projects like this one. So we screened colonies seeded in 12 96 well plates. So this is around 1,200 colonies. And following monoclonality validation and growth assessment, 35 best performing clones were selected, and antibody expression was validated by SDS-PAGE and ELISA. So the top 10 performing monoclones were expanded and cultured in fed batch conditions, and from these top 10 monoclones, 4 of them showed expression yields between 3 to almost 5 grams per liter. So the clones demonstrating long stability, making them suitable candidates for scale-up. So from the top 4 candidates, 1 of them was selected for scale-up and achieved excellent yields, around 8 grams per liter with optimized culture conditions. So we successfully developed a stable cell line capable of expressing the client's therapeutic antibody with more than 1,000 times increased yields compared to the transient transfection. If you remember, we started at 6 milligrams per liter during transient transfection, and we achieved almost 8 grams per liter starting after monoclonal selection. So in our pursuit of excellence in stable cell line development, it's crucial to recognize the key characteristics of a highly quality stable cell line and how Proteogenics leverages advanced techniques to guarantee success. So let's take a closer look. One of the first traits of high quality stable cell line is the high productivity that is translated by long-term expression stability, where the cell line must consistently produce the target protein of antibody through multiple passages. At Proteogenics, we develop monoclonal cell lines from selected pools, ensuring stability and high output through rigorous studies. The second essential trait, it is proof of monoclonality. And using the VIP system, we ensure automated monoclonal isolation through imaging. So VIPS tracks monoclonality, cell line survival, and growth, leading to selection of best stop candidates, which is a crucial step for downstream processes. Stable transgene integration is another essential trait of a high-quality cell line. So the gene of interest must be securely integrated into the genome to avoid loss or mutation. And our team uses stringent selection to ensure stable gene integration, preserving gene integrity and rigorous screening to remove aberrant clones, minimizing recombination risks and ensuring consistent production. Final product quality is highly important, as a developed cell line must produce a properly folded functional protein with optimal modifications and consistent production across batches to meet the quality standards of uniformity and efficacy. So our team conducts extensive assessment, including yield evaluation and SDS page under reduced and unreduced conditions to confirm protein integrity. We also use advanced analytical techniques like SEC-HPLC for purity, NanoDSA for stability, ELISA, SPR for antigen reactivity and binding kinetics, and mass spectrometry for protein integrity. So in addition, using high-quality expression and production media and monitoring across up to 90 generations, we ensure consistent yields and stable quality. Last but not least, safety and regulatory compliance is required in order to ensure integrity and traceability of the cell line. And Proteogenics performs a stringent quality control of the RCB, taking into account pathogen testing like mycoplasma and bacterial contamination assays during cell freezing and thawing to ensure pathogen-free cell banks. We also provide complete traceability. Every operation, including batch numbers and COAs, are fully documented for traceability. In addition, regulatory-ready documentation is provided, including monoclonality reports with visual documentation ready for regulatory submission. So our robust approach to stable cell line development combines advanced technologies with stringent quality controls, allowing us to meet the highest standards of productivity, stability, and safety. So with Proteogenics, you are assured of excellence at every step of the process. So in addition, using our proprietary CHO-K1 cell line, customer retains the ownership of the cell line at the end, with no royalty milestones or additional fees during the later phases of the project. So I hope I convinced you that outsourcing such a complex project like stable cell line development is the best choice to reduce costs, timelines, and risk of failure. And by partnering with a CRO like Proteogenics is a guarantee of excellence. Before I wrap up the talk and give the floor to my colleague Andrea for advanced instruments, I wanted to inform you about our special offer of a special end of the year offer consisting in a free audit and in silico analysis of your lead candidate before moving forward to the CLD project. So thank you everyone for your attention, and don't hesitate to ask your questions in the chat. Answer will be given during our Q&A session after Andrea's presentation. Ines?
So thank you, Laura, for the presentation. Now let me introduce Andrea Goff from Advanced Instruments. And also, as Laura say, if you have any question, don't hesitate to put that on the chat, and we will answer it on the Q&A question. Andrea, it's your turn.
Great. So thank you very much, Ines, for the introduction. Much appreciated. And thank you for inviting me to this very interesting webinar. So I'm Andrea Goff, Senior Director for the Celentim portfolio at Advanced Instruments, as Ines mentioned. I've been working for the company for some time now, and I have the great pleasure to talk you through some of the technologies that we have that will enhance navigating through the complexities of cell line development. But before I do that, I just wanted to give you a quick overview of Advanced Instruments. We have many different instruments in our solution offering, from osmolality testing all the way down to bacterial cultivation instruments, and also liquid volume calibration. So each of those have their own branding, Osmotec, Noxamat, and Artel. But what I'm going to talk to you today is very much about the Celentim arm of the business, and the instrumentation within that arm that really supports the efficiencies in cell line development. So starting straight from the beginning there, the biggest importance of cell line development over the last 10 plus years has very much been clonality, and it's such a critical factor for cell line development. And the reason for that and why it's important is because essentially the regulators have determined that it's really an essential requirement to ensure genetic homogeneity, and also alludes to the stability of the cell line. This picture we have here just outlines on day 0 how clonality is so important. Not only that, is monitoring that cell line from the original single cell throughout its life into a clone, and also maintaining that clone throughout its growth. It's really important, and having evidence of that is very important for the regulators. I won't talk about it in this slide deck today, but critical quality attributes of the protein or the drug you're trying to develop are also important, and we have solutions for that as well. But the very much the importance of this talk is to show you how protogenetics, excuse me, is using the VIPs to support their CLD. So if I move on to some traditional methods, limiting dilution has been used for a really long time in the industry. It's extremely low efficiency. You know, people are doing this by hand. It takes an awful long time for people to do this, and that ultimately leads to high costs, lots of consumables, lots of media. There's also a certain level of operator dependency. Whilst it seems like a very manual and easy process, cloning methods do actually require some skill, and it's quite easy to get it wrong. Cells will tend not to grow, or you'll get multiple cells in the well. It's just highly inefficient. Also, finally, it's the visual assessment of limiting dilution is highly subjective to the user, so it can create all sorts of errors. So this is where we introduce the Celentim portfolio. So we have in our range, we have the VIP single cell seeder, and that enables us on day 0 at point of dispensing to identify the single cell and make that seeding really accurate. Then from there, the Celentim portfolio offers whole well imaging, and that can be done on the VIPs, the VIPs seeder, or it also can be done on our Cellmetric instruments, our Cellmetric X instruments, which both of those monitor the clone from a single cell into a colony and really accurately do so, taking out all of the subjectivity of that. And then finally, I mentioned earlier that we have instrumentation that monitors the critical quality attributes, but we won't talk about this today. Moving back to the VIPs, as I mentioned, this is a really important part of the product and part of the CLD process, that it identifies a single cell in a well and determines that that well is clonal. When compared to methods like limiting dilution, the seeding efficiency is highly efficient. It essentially enables multiple wells of a 96 or 384 well plate to be dispensed in a short period of time, and you're not wasting your time with wells that haven't been populated. And how does it do this? So in that droplet that's dispensed into a well, we use AI-based algorithms to detect the single cell. And then that will essentially say to the user, if you have a single cell and we've detected a single cell, tick, yes, that's a cell. You don't need to worry about that. It's definitely there. If it's got multiple cells in it, it will identify those multiple cells or aggregates of cells and just mark that well as not clonal. And really, you don't have to waste your time with analysing that. Not only do we also do this for CHO, like Laura talked about in her processes, why CHO is so important in CLD. We do have the ability to also have high cell detection accuracy with a number of other cells, from HEC to iPSC, MSCs, hybridomas, and insect cells. You know, there's pros and cons with all of these different cell lines, but the instrument has the capability, using AI, to detect all of those cells to high accuracy. Now, I mentioned also the imaging side of things. This is really important. You know, the regulators don't just require a single cell, they require images of the clone being monitored into a clone. And that's what the whole well imaging on our VIPs and also our Cellmetric enable. Essentially, as I'm pointing out here, you can see the single cell. As it grows, the confluence is detected, and you can use those parameters to determine how healthy that cell is. And again, we can do it in 96 and 384 wells, as well as larger plate formats. Here you can see the whole well, the 96 well, and as you're expanding that clone, it can go into 48, 12, whatever your preference is, and the software will take out that subjectivity by measuring that confluence for you, so you can really monitor your clone. So finally, that's the beauty of the instrument here. As Laura mentioned in her talk, they are doing this process for their customers, and the VIP sits really nicely in this workflow to enable them to be more efficient for their customers. As was mentioned in Laura's presentation, the VIPs enables them to be 50% faster. It gives them higher precision and greater proof of clonality, which is really important for the regulators' compliance, and helping our clients and Proteogenics clients actually in their IND submissions. So that's it from me. Real whistle-stop tour of the VIPs and the whole well imaging. Thank you so much for listening, and I will hand back over to Proteogenics for any questions. Thank you.
So thank you, Andrea, for the presentation, which was very interesting. Interesting and very clear for us. So now we will try to answer all the questions on the chats. We had a lot of questions. That's very, very great. And so for the first question, I will start with Laura. What is the main reason for not use X large scale? Laura.
And yeah, I saw that. My mic was off. Okay, so can you hear me now? Yes, perfect. So there are several different reasons why HEC human embryonic kidney cells for scale-up were not that used for scale-up production. One of them would be lower productivity compared to the CHO cells. So CHO cells are preferred for large-scale biomanufacturing due to their ability to produce higher quantities of proteins, of antibodies. Another one would be the cost for media and cultivation. So HEC cells require extensive and complex growth media, which can sometimes significantly increase production costs, especially when they are scaled up. There would be also viral contamination risks because they have, as human cells, they have human-derived cells. They have a higher risk of carrying human viruses or endogenous retroviruses. And I would also mention cell fragility because they are more sensitive and fragile during scale-up culture conditions, like in bioreactors.
Okay, thank you for your answer. The second question going to be for Andrea. Can you expect on the regulation documentation that you provide through the VIPS system?
Yeah, so basically
you saw I mentioned in the presentation there that we have the VIPS drop and we also have the whole well imaging. So what the system does for you, the Studious software that we have, is it essentially on your request of whichever clones that you wish to include in documentation, it enables you to pull all of those images together in one timeline of that clone's life, essentially, into a report that we call the clonality report. And then that is the documentation that, say, Proteogenics can either give to their clients, and then you can give to your INDs, or basically give it to anyone that wants it, to be honest. It's definitely, as I mentioned in the talk, it's important not only to have a single cell image, but also track that cell throughout its life and growth into a colony. And that's what the report generates for you. If you've got 10 multiple clones, you select it and it will generate it for you.
Okay, excellent. Thank you very much. Another question for Laura. How does one go from a stable cell line that express a protein in high quantity and has been truly tested, but it's produced in a research environment through a GPM grade line?
Well, I think you need to contact CMO or CMDO to transfer your cell line with all the documentation and all the protocols and all the information on how you basically develop the cell line. And they will, and the cell line by itself, I suppose you have an RCB, so the research cell bank. So the CMO is going to develop the master cell bank and the working cell bank, and then they're going to do an extensive characterization of the cell line, and then they will basically continue with the manufacturing in GMP conditions.
Okay, great. Another for Laura. Can you explain more on how the developability assessment is done?
So we are running a thorough developability assessment on the heavy and light chain. So basically, we focus on the variable regions, so the heavy and light variable regions. So we are looking at post-translational modifications like glycosylation, oxidation, isomerization, and all other motifs that could be problematic for the manufacturing in GMP conditions, so large-scale manufacturing. We also look at hydrophobic patches and motifs that could be problematic for manufacturing afterwards, for the aggregation basically, and also the expression levels. So we will generate a report with all the problems that we identified, and then we will suggest solutions for that, and we will discuss, we will deliver you the report together with the solutions, and we will discuss with you the next steps of your project, basically.
Okay, thank you. Second question for Andrea. Can you explain on why your clonality photo are better than, say, upside?
Yep. Don't worry, I know what that means. Yeah. So we've essentially got a great whole well imaging. We've also got drop images. They are generally higher contrast images than our competitors in general. I am of course biased, but I have seen some of our competitors' images, and I think that we are providing higher quality images for our users. We've also been the market leader for that for some time now. You know, I also mentioned the multiple images that we provide on the new VIPS Pro. We also have multi-image plain images that we can provide. People can take the raw images if they want. But yes, essentially, it's difficult to answer that. I'm not obviously as really familiar with the UpSight, but I'm aware that they've been improving their images over time, and but we generally still get the feedback that our images are the best in the market, just because of that high contrast and the ability to see that single cell very clearly.
Thank you. For Laura, is your vector IP free? What selection maker do you use?
So our vectors, yes, are IP-free. And our CHOR-K1 cells are used with methionine sulfoximine selection as a part of glutamine synthetase selection system. That's about all the detail I can give you right now, but really, if you want to have more information, just don't hesitate to book a call with me.
Okay. We'll send maybe the link at the end of the
Right. Sure.
webinar. Maybe another question for Laura. Do you clone HC and LC in different vector on the same vector? If first, do you optimize and assess different vector ratio?
Very good question. So that depends on the biomolecule that you want to express. If you're looking at the classical IgG, we would clone both heavy and light chains of the same vector. If we are looking at bispecifics, then it will depend on the format of the bispecific.
Okay. Maybe for Andrea, does documentation of VIPs is in compliance with FDA documentation, I think?
Oh, yes. So that's a difficult one because the FDA never actually specifically says what everyone needs to conform to.
Unfortunately.
Yes, they're very vague.
Yes.
But the reason for that is to allow people the bandwidth to produce what they think is appropriate for their process. But generally speaking, we have information that they are used in INDs. We actually have an application note of another developer of ours that is using them in an IND. So I think there's no actual strict compliance that the FDA specify, but the more information you give them, the better, essentially, and the clearer the images of the single cell that then develops into a clone, and any debris around that you can identify will help the FDA just have that confidence in that clonality right from the start. So it's a good question, and I wish the FDA did give guidance on what everyone should do, but they don't. All I can say is that they have definitely been used in submissions.
Okay, perfect. Thank you very much.
So maybe they can
Laura?
just continue, but because there was another similar question, well, to what extent does the development of a stable cell line needs to be documented? So again, it's not very clear, but as much information you have, it is always better to have as much information as you can from the beginning. So starting with the cell line, so the origin of the cell line, and during all the process until the end of each and every step. So yeah. Thank you.
Okay, so you speak about the process, so I think the question is good for that. So how quickly can Proteogenics deliver a stable cell line from start to finish?
Right. So this is another very interesting question, and it depends on the project, to be honest. So the quickest we can do is 10 weeks. There will be no-goes. There will be, I mean, this is really the quickest that we can go from having the optimized sequence, developing pools, doing the monoclonal selection with VIPs, and then just having the cell at the end. Then we do offer several different options, as I mentioned before. So the developability assessment, expression, transient expression of the variants that we will identify to have an idea of aggregation, stability, yields, and everything. And once we have the best candidate, we'll go forward to the pool development. Once we've identified the top 3 pools, we usually do fed batch expression tests and purification tests to have enough material to basically analyze the physicochemical properties of the antibodies. And then this will allow us to select the best pool to move forward with the monoclonal selection, the selection of monoclones. And again, as you've seen in the case study, once we've selected the top 3, 5, 10 monoclones, they will be multiplied and expressed in fed batch conditions, and again purified and characterized to have an idea of the top monoclones that we can deliver to the customer, or even 5, depends on the project. And then let's not forget the RCB stability studies that can go from 30 to 90 generations, and this will add additional time to the project. So everything you can customize everything. So starting from 10 weeks up to, I would say, 3 to 4 months, depending on the stability study duration that you would like to do.
Okay, thank you. And any specific supplements used during the single cell line cloning planning process?
That's another good question, and I will not be able to give you the answer now.
Okay, it's happening sometimes. Maybe for Andrea, questions. Is necessary to calibrate the VIPS Pro to cell size, and how does it alter the proliferation of cell after sending?
Sorry, I hadn't unmuted myself. Yes, so absolutely not. So the VIPS Pro uses AI to detect the cells. So we've actually fed the instruments Studious software with loads and loads and loads of images for training. So we've done that on all of those cell lines that I listed. So you don't necessarily, even though there is a parameter in there, if you wanted to adjust it, we've essentially trained it using AI. So it detects the cells as they're dispensed and confirms the presence of a single cell by that AI, artificial intelligence. And then to answer the second part of the question, how does it alter the proliferation of cells after seeding? I'm not quite sure by that question, but essentially after seeding, the cells are continued to be monitored by AI algorithms. So we essentially have in the whole well where the cells are dividing and growing, we have a confluence overlay that's determined by, again, AI, and that maps the cells. And then what that means is you don't need to change any parameters in terms of like the cell size for detection. It's literally using AI to determine where the cells are, and then it gives you that confluence output. So hopefully that answered that question. If not, feel free to stick another question in there.
Okay, thank you. Maybe for Laura, how are the in silo assessments, QC, and validation step adapted for no antibodies biologics?
Very good question again. So the in silico assessment is going to have a look at the same parameters, so glycosylation, oxidation, deamidation, and we'll give a report on that. It's going to have also, we're going to have an idea of hydrophobic patches, because I suppose that even antibodies are also proteins. So all these, the characteristics that we are looking at for antibodies are very important for general proteins also. So QC validation would be the same. So I would say HPLC for aggregation, reducing and non-reducing SDS page, nano DSF to have an idea of the thermostability of your protein, especially if it's a biosimilar protein, for example, and you would like to compare it with the molecules that are already on the market. But this is a little bit more complex than that. There is a huge panel of analysis that need to be done if we're talking about biosimilar proteins. So I would say that all the steps that we're looking at on the antibody side can be very, very well applied to the protein spectrum as well.
Okay, thank you. We are running out of time, so maybe a last question for maybe Laura. Which integration system are you using? Means it's random or site specific? It's totally.
With this random integration system.
Okay. So okay. So I think we have a lot of questions. We don't answer it, but actually we the webinar is almost finished. So we'll take time after the webinar to answer all the questions. We take all the questions, so we will take the time to answer them. And I will want to thank you, Andrea and Laura, for the presentation and the answer for this webinar. And so I hope you have enjoyed